biomedical engineering uses. So as I said, like it has a wide range of applications. So biofuels, for example, obviously bioplastics. So with biofuels, the PHA can be just synthesized into butanol and propanol, which are biofuels. So a problem that we're trying to solve is how can the PHA production be increased on a larger scale to make more biodegradable products? Because at the end of the day, it's a product made from a microbial host. Obviously, microbes are not big. So how can we do that? So just a quick introduction to what the polyhydroxide alkanate actually is. It has this general structure. They all have this general structure. And these are kind of the differences between them. So what really differentiates all the polyhydroxide alkanates is their chain length. So you have the short chain length of polyhydroxide alkanate, and then you have the middle chain length. So in our microbe, the Pseudomonas putida, we have a middle chain length polyhydroxide alkanate. So these PHAs are polyoxiders composed of linked hydroxide alkanate units. So they both have a carboxylic acid, and they have a carboxylic acid group. So these are just monomers formed into long-chain polymers. So how are they produced in the bacterium? So they're naturally produced as a stress response, but they're produced as water-insoluble granules. So that's also why it's such a good product, because since it doesn't dissolve in water, it can be used for a wide range of applications. So in the Pseudomonas putida, you have two genes that encode the enzyme for the polymerization of this PHA. So you have the PHAC1 and PHAC2, which again encodes the enzyme that catalyzes the polymerization of the monomers into the polymers. So as you can see in the bottom, it goes monomer to polymer. But then you have another gene, the PHAC gene, that encodes a depolymerizer in this sort of bacterium. So the PHAC is responsible for breaking down the polymers back into monomers, just to reuse again in their environment. So you have to take in mind that the PHA is formed in a nitrogen limited but carbon rich environment. So it's not so much that the environment is carbon rich as in having more carbon resources, but it's about the ratio. So the nitrogen is limited, so naturally the carbon will be higher than the nitrogen. So it forms this sort of granule in response to that condition. But the PHAC, again, it breaks it down, so it can reuse. So what challenges do we have from this PHA production? So although you can see here, we have a good ratio of the PHA granule to the actual putida cell, the PHAC enzyme or gene sort of limits how much PHA or bioplastic we can actually collect. So because the bacteria naturally degrade the PHA back into monomers due to the PHAC gene, then we need to find a way to resolve or sort of knock out this PHAC gene. So hopefully we can increase how much polymer we can actually collect. So how did we do that? So we tried to knock out the PHAC gene in hopes to, again, eliminate these depolymerase activity and the recycling process overall to increase our yield. And we utilized the CAS system. So I want to highlight the CAS system a little bit, just because it's a relatively new technology, but it has, it's a new method that can be used for a very efficient sort of experiment. So this CAS technology, it's a CRISPR-associated transposase. So it's similar to the CRISPR-Cas9 system. So it's a gene editing system, but unlike the CRISPR-Cas9, it doesn't create that double-stranded break in the DNA. So this utilizes a CRISPR guide RNA-guided protein but it utilizes a transposon to sort of insert the DNA fragment without creating that double-stranded break in the DNA. So when you're making your experiment or you're conducting your experiment, you can utilize this instead of the CRISPR-Cas9 to make, for example, only one plasmid, whereas with the CRISPR-Cas9, you will have to make two, one for the cut and one for the repair. But with this, you don't have to do that, making your experiment easier and faster. So we designed our plasmid and we inserted antibiotic-resistant genes. So we utilized kanamycin as a marker. And then we tried to create a frameshift mutation to knock out our PJC gene. And again, we utilized a Cas system to do that. So we used the Golden Gate plasmid assembly to sort of make our plasmid just because you can organize multiple DNA pieces simultaneously. So after that, we used the transformation, which is basically you shock the cells so your plasmid can go in there and then you recover them. Then after that, we inoculated them and we let them grow. So although we can have our colonies grow, and if our colonies grow, they have our plasmid. But the question is, do they have our mutant? Do they actually knock out the PHAC gene? So through that, we did the colony PCR, which is just doing amplifying the DNA. And then we ran a gel electrophoresis to sort of check whether the colonies had our gene or the lack of our gene. So as you can see here in this gel electrophoresis, you have this sort of top band, and then you have this bottom band. So one thing I want to emphasize is like our wild type, which is the sort of like the original type, it's shorter than the PHAC knockout. So why is that? That's because the Cas inserted the DNA fragment into the plasmid, so it would naturally be a bigger size, or it will have higher molecular weight. So due to the cargo inserted utilizing the Cas system, the molecular weight was higher in the PHAC knockout. So as you can see here, the PHAC knockout is around 1,000 base pairs, whereas the wild type is around 626 base pairs. And that basically confirms our experiment, and it confirms the fact that it does in fact have the PHAC knockout. So again, the Cas-mediated PHAC knockout appears to have been successful. So the PHAC gene has been disrupted, as indicated by this gel electrophoresis. So our PHAC knockout strain, again, was successfully interrupted. So this knockout is expected to eliminate the depolymerization activity in this system. So with that, we hope to increase the PHA yield, and it will be used for industrial purposes, so it will generate more PHA. So our future directions is we'll quantify and compare PHA production between the PHAC knockout and the wild type to actually assess the impact of the gene disruption. Because a problem we might encounter is through knocking out this PHAC at the end of the day provides balance to the cell, so it counteracts with the activity of the polymerase. So if you have an overexpression of these genes, that will sort of add a stress to the cell, and it could cause a metabolic burden, which would have the opposite effect of what we actually want. So with that, I just want to thank everyone for being here again, and I really want to thank my mentor, Coxie Lee and Fuzhong Zhang for this opportunity and for the opportunity to do this amazing work. So without further ado, do any of you have any questions?
The natural role of pH?
Yeah, so naturally the putida or bacteria in general they produce this PHA as a stress response. So in an environment that's nitrogen limited but carbon rich, they use this as sort of a hibernation purpose. So they will store this PHA sort of for later use and for energy or a carbon resource so they could have later. But then they break it back down whenever they have that carbon deficiency. Other questions?
I had a related question. I think you stress response, and I think you molecule.
Yes.
That it's. So where do these bacteria naturally exist?
So
In environments where there's.
So, I mean, the Pseudomonas bacterium naturally exists in the soil, but in an industrial sort of setting, you can feed it carbon. And the good thing with Pseudomonas putida is it can be fed a lot of things. So it can be fed waste or glycerol, sugar, whatever. So it makes it a very good sort of micro for these industrial purposes. Other questions?
Questions on Zoom? Other questions? Thank you, Valerie. Valerie.
Thank you.
Our next student is, you can do it. It's Irene Dumitriu from Georgia Tech, from the Foston Lab.
Sorry. Yeah.
Hi, everyone. My name's Irene Dumitriu, and this summer I had the incredible opportunity to work in the Foston Lab, where I worked on the optimization of polydopamine in a biopolymer composite for increased biocompatibility. So there's an increasingly urgent demand for sustainable biodegradable materials, and this is because our environment is being polluted by petroleum-based plastics. So a plastic alternative is our biopolymer composite. Now, what is a composite? A composite is a material that is comprised of two or more individual materials that have differing properties, and when they are mixed together, they make a composite that hopefully has superior properties compared to those individual materials. Our specific biopolymer composite is made of cellulose nanocrystals, or CNCs, and soy protein isolate, also known as SPI. These two materials are actually naturally derived or bioderived. So CNCs, they can come from any cellulosic biomass, and SPI, or soy protein isolate, can come from the soybean plant. So why did we pick cellulose nanocrystals? So cellulose nanocrystals have this incredible property where they have a Young's modulus, or a stiffness, that is comparable to steel. So they're a very, very rigid and stiff material. So when we add these really stiff nanocrystal rods into a polymer matrix made out of soy protein isolate, they create this biopolymer composite that is now reinforced because of these rigid, stiff material. However, our interactions between these two materials is not as strong as we would like them. So our biopolymer composite doesn't have a high enough tensile strength to compete with these plastics. So we have to further enhance the interactions between our two materials. And we do this by adding in a third polymer. This polymer is called polydopamine, or PDA. And polydopamine is a bio-inspired polymer that comes from mussels. Mussels have a protein that allows them to adhere to practically the surface of anything. So you'll commonly find mussels like at the bottom of boats and like adhere to them, and that's because they have an adhesive protein. So PDA has a very similar structure to this muscle protein. And our PDA acts as an adhesive coating. So we coat our cellulose nanocrystals in our PDA so that we have enhanced hydrogen bonding and interactions between these two materials to create a mechanically enhanced biopolymer composite. However, our lab has found in previous studies that even though it's a bio-inspired polymer, excessive PDA can actually introduce toxicity into our material, which is not what we desire. So this specific study studies the optimization of our PDA coating. So we want to try to find the lowest thickness possible while also maintaining those superior mechanical properties that we had before. For materials and methods, we use a rapid deposition method where our PDA is coated onto our CNC surface via dopamine polymerization triggered by a copper sulfate and hydrogen catalyst. But even before we can get to this rapid deposition method, we have to first start with creating our cellulose nanocrystals. Our CNCs, they actually come from wood pulp. So we take wood pulp, we grind it into a powder, and we use a 64% weight percent sulfuric acid to conduct acid hydrolysis. So the goal here is with acid hydrolysis, we want to separate our amorphous and crystalline regions from each other because we want to really isolate that crystalline region to create our cellulose nanocrystals. Once we've created our CNCs, we can now move on into our coding. And with this coding, we have three variables that we're really trying to target and optimize. And the first variable that we want to optimize and change and play around with is the amount of catalyst that we have. The second variable that we want to change is the initial amount of dopamine that we add into our system. So we actually don't start with polydopamine. We actually start with, we just start with the monomer, which is called dopamine, or DA. And our dopamine via that catalyst gets oxidized, and once it's oxidized, it can then form our PDA, our polydopamine polymer, and it can hydrogen bond with our cellulose to form our PDA CNC system. So our last variable that we also are optimizing is actually the pH. With the pH, we can try to control our reaction kinetics, and with this, we can try to optimize the actual coating thickness. Once we have our PDA CNC solutions, we can now move on into our last step, which is creating a nanocomposite film. So here we take our PDA CNC solutions and we add them into our soy protein isolate matrix, or SPI, and we solvent cast them to create our nanocomposite films that we can then further analyze and test. But before we move on to testing these films, we have to first verify that we have a proof of coating. And we do this with AFM. So I have 3 AFM images here. One is just our CNCs by themselves without any PDA coating. Second image is just PDA, no CNCs. Our third image is a formulation of 2 times DA and an 8.5 pH, and that's our PDA coated CNC. As you can see, with just CNCs only, they have a rod-like structure here. PDA is more of a spherical globular structure, and that's because PDA can also bind with itself. So commonly you'll find a lot of like PDA aggregates, which is why this step is so important, because we need to verify that our PDA is not only binding to itself as it can, but it's binding to these nanocrystals, which in our third image, you can see that these structures are more of a combination or an in-between between these 2 images. So we've verified, at least visually, that we do have an adhesion going on. We can also use FTIR to verify this proof. With FTIR, we just have various formulations here, and we have our baseline CNC, no PDA, and we just have our 2 different pHs that we tested, which was 8.5 and 10. And it's a little hard to see, but there is a peak here, like that follows through all of these that is not present in our CNC. It's very small, but that peak represents our carbon-to-carbon double bond that's present only in dopamine, and it's non-existent on our cellulose. So here we've also verified that we have a PDA coating on top of our CNCs. With this verification, we can then move on to actually optimizing this coating thickness, and we use dynamic light scattering, or DLS. And dynamic light scattering essentially just allows us to use how light scatters off of our particles to see what the actual size of our particles are. We're hypothesizing and assuming that a larger particle size means a thicker coating of our PDA. So here, with increasing DA concentration, we can see that there's an overall trend that leads to an increased particle size. With our increasing copper, our catalyst concentration, there only really is that overall trend with that pH 10. Here in our pH 8.5 is not as strong as a trend. And then with our two different pHs of 8.5 and pH 10, there's not a huge difference between them except for our 2 times copper sulfate. Moving to the next graph, we have our zeta potential. So DLS not only allows us to see the size of our particles, but allows us to see the charge of our particles as well. Zeta potential, essentially, again, just the charge of our particles. PDA is negatively charged. So we're assuming that a lower charge or a higher negativity means that there is more coverage of our PDA coating onto those cellulose nanocrystals. Here for the trend, consistently over all of our different samples, we have a higher pH, consistently over our samples with a higher pH of pH 10, we have a more negative potential. And so we're assuming that a higher pH leads to a more coverage. Our PDA is more uniform and more covered onto our CNCs. Now, once we've created all these solutions and we've verified them and we've also just optimized them, we can move on to our nanocomposite films. We have 3 films here. We have our SPI control, which is just your soy protein isolate matrix, no PDA, no CNCs. And then we have 2 samples here. They are our 2 times copper sulfate and then 0.25 copper sulfate. We picked these 2 because there was a difference in thickness. So our goal here is to make sure that even though there's a difference in our coating thickness, we want to make sure that our mechanical properties remain consistent despite this difference. So we use a tensile meter to test the tensile strength and our mechanical properties. I have a stress strain graph over here. So you can see that there is quite a big difference, at least in the curves, for our control versus our 2 samples. And we have our ultimate tensile strength and we have our Young's modulus over here. So in our ultimate tensile strength, there's a significant difference between our SPI control and our 2 samples, which is denoted by these asterisks. And that means that our PDA CNCs did indeed increase the ultimate tensile strength of our material, which is great. And we also see that there is a non-significant difference between our 2 samples, which is also what we were looking for because we want to make sure that despite that thickness, we have a similar tensile strength with our composites. And then for Young's modulus, there was no significant difference between any of our samples. So for this study, what we've essentially learned is that increasing the pH of our material, so a pH of 10 versus pH of 8.5, allowed us to have a more negative zeta potential or more PDA coverage. And then increasing our catalyst concentration, specifically at that pH of 10 over here, allowed us to have a thicker coating or a larger particle size. And for our ultimate tensile strength, we were able to maintain our mechanical properties between our 2 samples while having a difference in coating thickness. And in summary, to achieve the lowest but also most uniform PDA coating, we want to increase that pH, but we want to decrease the amount of catalyst that we add into the system. So now we have an optimized PDA coating that allows us to better compete with our petroleum-based plastics and mechanical performance while also maintaining our sustainability and improving our biocompatibility. And I want to thank my lab this summer. I had a great experience, and thank you for my bench mentor, my Bayer mentor. I learned a lot. So thank you. Any questions?
Getting to poly-based.
I know for our specific experiment, compared to our previous studies, these specific values are lower than previous values, but that might just be like a method difference, a batch difference in our CNCs. So my specific data is like lower, but we did have previous data that had higher tensile strength that was a little more comparable. But this is just my specific data. I think I remember maybe like tensile strength wise, like I think plastics was like 110 MPa, and we were seeing definitely lower than that, but more like, so like 30, but hopefully getting closer.
Okay. Thank you.
Yeah.
So how?
Can you verify like what's economically viable?
Like,
I'm not sure about economically. The problem with getting something that's like bio-derived, I think there's a lot more processing that you have to do. So like for us, we do have to process, like we don't make CNCs, like we do have to process them from like a natural thing to like get that. So I'm sure there's like less economic value, but we also are working in like a circular economy. So like when we take something from nature, these can go straight back into nature. So don't know if that answers your question, but maybe more expensive, but better for the environment. Other questions? Okay, thank you.
Our next presenter is Sowon Kang from the Pathak Lab.
Okay. Hi, everyone. Thank you guys for coming here. My name is Sowon Kang. I'm a rising senior at the University of Texas in Austin, majoring in biomedical engineering. And this summer, I had a chance to work with Dr. Amit Pathak's lab, mentored by Jade. And I studied how hypoxia and collagen density regulate the breast tumor cell invasion. So starting off with some introduction. So this might be a small image, but tumor microenvironment is a dynamic network of cells, biomolecules, and structural components that kind of surrounds the tumor and provides like biochemical and mechanical cues to the tumor itself. And there are different types of biomarkers of tumor microenvironment, but for our study purposes, we're focusing on two of them. One is hypoxia, which refers to lack of oxygen, and two is the extracellular matrix stiffness. So hypoxia, which refers to lack of oxygen, so it's very common in most solid tumors because inside of a tumor, there is like a lack of blood vessels, so it prevents the delivery of oxygen. So it's very common in most tumors, hypoxia is. And previous research have shown that hypoxia is associated with like increased invasion, metastatic initiating capacity, and a resistant phenotype. Like there was a study in 2019 that showed that cells that are hypoxic are 4 times more likely to circulate the blood and metastasize to other regions compared to like regular cells in cancer. And another point that I was focusing on was the extracellular matrix stiffness. So previous studies from our lab showed that cells primed on stiff matrices compared to the cells primed on the soft matrix show a faster migration and a greater invasion distance, as well as a faster migration speed. So this also shows that previous research have shown that if cells are in a stiffer environment, they're more likely to invade, especially for cancer cells. So our question is, what if we combine those two, hypoxia and ECM stiffness? So the recent study from our lab have shown that if you grow the cells in like a stiff and hypoxic environment, it leads to the greater invasion and more metastatic potential. So we wanted to explore that question even further, which leads to our research question, which is, how do hypoxia and collagen density affect the breast tumor invasion? And our hypothesis were that hypoxia and collagen density have independent influences on tumor invasion, and these influences may act in a cooperative or a competitive manner. So moving on to my methods. So I grew 2 cell lines this summer, 4T1 and BT549. 4T1s are the breast cancer, mouse breast cancer cells, and BT549s are the human version of the breast cancer. So I grew these 2 different cell lines and then created a spheroid, which is like a cluster of cancer cells. And I plated them on a spheroid plate and then incubated them at different oxygen concentration, 1, 5, or 20%. For reference, 20% is considered as like normoxia, the normal oxygen concentration, and anything below that is considered as hypoxia for our study. So after incubating them, the spheroids were formed, and I plated them on a collagen gel at 3 different concentrations, 2, 3, and 4, to vary the stiffness. And then after 24 hours of more incubation at the same oxygen concentration, I did a live cell imaging using the microscope for 48 hours. So 2 days imaging of how the tumors grow after that. And then after that, I did a trypsin assay, which is basically adding trypsin to the cells, waiting like an hour for the cells to detach and ball up. And after that, I imaged the cells again. So that was my method. And moving on to the results. So these are the images I took. It's like a sample image of the 4T1, the mouse cancer at 5% priming. And then I traced the area over after every 12 hours to see how much they have grown over time. And as you guys can see the difference, the 2 mg/ml is the greatest, and the 4 mg/ml did not grow as much compared to 2 mg/ml. So it kind of tells that higher collagen density in general kind of impedes the tumor invasion for this case, and this was true for the BT549 cells as well. So after I traced this area, I quantified them and then generated these 2 graphs for 4T1 change in area and the BT549 change in area. And from here, one thing to note was that 5% hypoxia priming was the most sensitive to collagen density for both cell lines. When I say sensitivity, I mean the difference between the 2 mg/ml and the 4 mg/ml. So if you like connect these 2 red dots and then draw a line and find a slope, that would be the sensitivity. So from this graph, the 5% hypoxia had a greatest sensitivity for 4T1, and that was also true for the BT549 cells as well. And another thing I found was that for higher concentrations, like 3 mg/ml and 4 mg/ml, the 20%, the blue line, was greater, had a greater invasion in area, but only for 2 mg/ml, 5% was the greatest, which was pretty interesting. And this tells us that maybe something is going on at the 5%, 2 mg/ml hypoxia in terms of tumor invasion. So we wanted to explore this further, and I moved on to the trypsin assay, like I mentioned earlier. So when you add a trypsin, the cells ball up, and I tracked the displacement of the beads. So when I was creating collagens, I added the beads to the collagen, and when I imaged the tumors, I also imaged the location of the beads, and then created a vector map that shows the movement of the beads before and after trypsin. And then I found the average of these beads displacement data. And then these are the graphs of the beads displacement for 4T1 and BT549. And then first of all, one thing I noticed was that in general, 4T1 cells had a greater displacement compared to the BT549 cells. They also had a greater increase in area as well. So they had more displacement, this cell line, compared to the BT549 cell line. And another thing I noticed is that at 20% is also similar. At the 5% hypoxia was greatest had a greatest beads displacement for 2 mg/ml for both for both cell lines. That's the red graph here. For 4T1, the 5% hypoxia had a greatest beads displacement for 4T1 cell. And also for BT549, the 5% hypoxia had a greatest beads displacement as well. Again, this is a similar trend that we saw from the area. So this leads us to think that maybe hypoxia is affecting the invasion in some density-dependent manner, because this trend is only occurring at 2 mg/ml at a lowest collagen density. And the last thing I did was looking at the traction force. So traction force is the measure of the force that the cells exert on the matrix as they move out. And the way to measure this was I used the beads displacement data and then the Young's modulus that were calculated using AFM. So then you can quantify the traction force on image day. So when I quantified the traction force, I found this trend that, first of all, again, the 4T1s had a greater traction force compared to the BT549 cells. And then another thing was that this was interesting because 20% normoxia had a greatest traction force for almost all densities for both cell lines. And then this kind of relates back to what the point I was talking earlier, because for the area the change in area and the base displacement, the 5% had a greatest change in area and the base displacement, but 20% had a highest traction force. And 5% traction force was in the middle for the 2 mg per mil data. And this tells us that there may be some balance of forces that are going on at 2 mg per mil, 5%, that causes this increase in area and increase in base displacement, even though it has like the medium traction force in this case. So that was pretty much what I found overall. And in conclusion, the hypoxia priming and collagen density regulate the metastatic potential in a complex cell line specific manner. Like I was saying earlier, 14-1 and BT549 had similar trend, but also slightly different. Like 14-1 cells generally had a higher increase in area, base displacement, and traction force compared to the BT549. Maybe the difference between human versus mouse cancer cell line. And then increasing collagen density led to reduced invasion in both of the cell lines, in both the change in area and the base displacement. And one possible reason for this is that if you have a greater collagen concentration, you're in a stiffer environment, and that may physically restrict the cells by reducing the pore size and restricting the movement of the tumor. So that could explain why like the cells grow much faster and more in 2 mg per mil, whereas not as much in 4 mg per mil. And then we also hypothesized that another thing we found was that each cell line exhibit different sensitivity to collagen density under hypoxic conditions. And this could be due to the interplay between cell migration forces. I was talking earlier that there are 3 different types of cell migration forces: protrusion, adhesion, and contraction. And the balance of these forces may act differently at different oxygen level. Like something might be activated or deactivated at 5% hypoxia at 2 mg per mil, which could be why this is occurring. This phenomenon that we observe is occurring at this level. And then lastly, while most studies on cell migration previously have focused on normoxic conditions, this kind of shows the need to study these cell migration at a hypoxic condition, because it's also physiologically relevant. A lot of cells in our body are operating under 20% oxygen level. So this study will be relevant, and more study needs to look at how cancer cells are working in a hypoxic condition. And then for future work, to study like how these activations of forces work, we could potentially perform immunostaining of different proteins like actin, paxillin, or like GLUT to see which forces are actually activated or upregulated, downregulated at different hypoxic conditions. And yes, I would like to thank Pathak Lab. Thank you, the lab members who all came to support me. Thank you, CEMB and the WashU McKelvey School of Engineering. And thank you.
Looked at.
Yes, so I did not look at it, but I saw that previous studies have looked at the cells at 20% oxygen level, not hypoxia level. And I was saying that that's not physiologically relevant because cells in our body do not operate at those levels.
So what do you think?
That might, I think there would be, especially for cancer cells, there would be, because that will help them with their survival and allow them to metastasize more. So if those phenomena occur at cancer cells, and it kind of goes back to what I was talking about with like the cell migration forces, my hypothesis is that certain forces are activated at certain oxygen level while other forces are deactivated at different oxygen level. And that might be causing those phenomena that we're observing at those cells. Other questions?
Our next presenter is Gbenesira Linus-Barinwa from the WashU Lab.
All right, hi, my name is Gbenesira. I'm a rising sophomore at St. Louis University, and for the past several weeks, I've been working under Haley Morrison in the WashU lab down in Rebstock Hall, and they've been exploring redox strategies within purple non-sulfur bacteria. So I'll be giving a brief overview about what I've done in their lab and what we've learned so far. So with a little background on PNSB and how we can contribute that to our real world issues, we know that as climate change has worsened and carbon emissions have been on the rise, there's also been a rise of how we could find sustainable solutions for our environment. And so purple non-sulfur bacteria, or PNSB, is one of our model organisms because it is it shows flexibility in metabolic systems. So the image is pretty small, but over here, this is the cell or the bacteria, and it holds a lot of pathways within it. So it's phototrophic, so it can do photosynthesis like it can in plants. It also holds a carbon fixation process right here. It does PHA production as well. So that's polyhydroxyconate, which helps with bioplastic production. It also has nitrogen fixation as well, and as well as extracellular electron transfer. And with a little bit of background behind that is that it's shown to transfer or receive electrons from external acceptors, so it could cycle through electrons within it and outside of itself. And with that, and all the processes within it, we've been bioengineering strains to knock out genes to see if the PNSB bacteria could still function and can still maintain homeostasis with like one gene knocked out. So, or one of like each gene knocked out. And hopefully with our studies, we could incorporate that by enhancing microbes to recycle things like carbon or electrons and build on to other sustainable products. So over here is the table of strains that we've used. And on the side is their purposes. So our wild type Pseudomonas is the control. And then we have our other modifications here. So have our Pio, which is for EET loss. We have our Fosi, which inhibits PHA loss. We have Rubisco, which inhibits carbon. And we have Nife, which inhibits nitrogen as well. And then we also have some more bioengineered strains at the bottom, which goes with fatty acid production. And so with the methodology that we've used throughout the weeks, which took a pretty good time for each, one of the first things we did was culturing tubes. So we cultured tubes inside an anaerobic chamber where there were 3 samples of each modification. Some were fed acetate and some just depended on the hydrogen within it. And so we dispensed each sample, we fed some acetate, and then we took it out the chamber, we capped them, and then we gassed them to make sure that they were strictly under anaerobic growth and there wasn't leftover like oxygen to make sure our experiment was accurate. And we left them in a warm room under around 30 degrees Celsius so they can grow under a white fluorescent light. And with that, we measured their initial OD or optical density. So that way, throughout time, until it reached stationary, we're measuring how much or how long it would take for cells to grow within the tubes. We also used a measurement called gas chromatography, which is when we took gas before and after the tubes grew, and we would measure like what type of gases were within it and how much was in it. So that way we could also check if there was any gas leaks along the way of the experiment. So we measured the carbon dioxide, hydrogen production, and nitrogen as well. We also did some gene cloning for constructs of our other modifications. So with that, we used hi-fi assembly, which was a way of like constructing the wanted genes that we needed for our modifications. And then we used those constructs and put them inside E. coli through electroporation. So electroporation is like a process of shocking the E. coli so that way DNA could go inside the gene. And we used conjugation, so the genes could like pass through each other, or the plasmids could pass through each other, through the bacteria within each sample. And then we made sure that there was no mutations or contaminations by verifying through PCR. And then we also did extractions, more so in relation to our bottom Taiwan modifications, but through that we didn't see any fatty acid production. So that was like a little side data that we had on the side. And at the bottom right here, we can see the graphs, and our graphs were pretty tame. It showed like a pretty precise growth throughout all of our strains. So up here, this goes with our top half of strains. Under our 1 millimolar acetate, all the mutants and wild type grew comparable densities, so they were all pretty fine. And same with our hydrogen gas, all the mutants were shown to grow to a similar final density. You can see our NIFA had a bit of a lag phase over here, but it still managed to go up here. That could indicate either a growth effect or just how it reacts to the hydrogen growth itself. And then with our fatty acid ethyl esters, they showed like similar results as well, where all the strains still reached the comparable densities at the end, and same with under hydrogen gas. And so with that, we can see that our final conclusion is that our PNSB is able to still manage homeostasis even if each gene is knocked out, and that can contribute to studies where like if there was something that needed PNSB for a specific process within it, like carbon fixation, for example, genes could be knocked out and PNSB would be for it to cycle through that carbon, or same with like extracellular electron transfer, where like other genes could be knocked out, and hopefully PNSB could still stay just as fine with any type of research or experiment that would just focus on that one pathway. So PNSB is shown to have and be a great model organism to just like focus on certain pathways to produce or like contribute to any type of issue and research along the way. And with that, I would like to thank my mentors again and my other lab mates at Rebstock Hall for helping me with the process of figuring out the tools there. And my PI, Arpita Bose, for still giving me some advice when it came to just presenting and having and using and asking questions about like the lab in general, and using resources around me to help with putting together our research project. And so, yeah. Any questions? Oh, you had a question? Yeah, I've never heard of a microbe that could hold like so much. Like it seems like a super microbe in my eyes. Like it could do like almost anything. Like it could almost like in my eyes it would seem like it could survive like almost anywhere. Like it could do photosynthesis and also like nitrogen fixation on the side, and how it could like cycle from one pathway and another just to like keep it going. I don't know, it almost seems like invincible. Like that did surprise me when I first heard about it, because I've never heard of a microbe that could hold like, what is it, 5 different pathways at once. It's like it never wants to die.
Or I was, yeah, that's.
Yeah, I think that would be a great way to look at it further in the future, since it did take some time and a couple fails to kind of make sure we could accurately get the results for every pathway being knocked out. That did take some time back, but I would sure love to see like how it would look like if like everything but one pathway was left, to make sure that it could still sustain that much. You could find this naturally in like aquatic environments, or some soil environments as well, where it's like thriving off of well, it kind of like answers that with like the processes you'd see in like plants. Like it has like nitrogen fixation and it's phototrophic as well. So you can find it around plants, and it's also helped to contribute in some plant growth as well, from what I've seen in studies. It's just like another symbiotic microbe that like latches onto plants or around it that could also help it grow.
Our next presenter is Jasmine Brown from the Wagon Seal Lab.
Hi everyone, my name is Jasmine Brown. I'm a rising senior at Georgia Institute of Technology, and this summer I spent my time in the Wagon Seal Lab. The lab focuses on vascular mechanics, and I was researching how elastin degradation limits aortic function by imposing a mechanical ceiling on contractile and dilatory responses. So just for some background in this research area, so vascular disorders affect millions of people worldwide, and they can be caused by several things like genetic factors, lifestyle changes, and then also environmental factors as well. One such condition is ascending thoracic aortic aneurysm, also known as ATAA, and this is characterized by an abnormal dilation or ballooning of the ascending aorta. As we can see in this image here, this is a normal mouse ascending aorta versus ATAA. We can kind of see that like abnormal dilation and increased diameter of the ascending aorta. And so this can be caused by several things like inflammation in the aorta, as well as loss of smooth muscle cells, and then degradation of extracellular matrix proteins like collagen and elastin that are useful for helping that vessel kind of like stretch and recoil when blood is pumped through the heart. Currently, the treatments that are used for ATA are trying to slow the disease progression. So for example, some can be like blood pressure medications that are trying to limit hypertension and then reduce pressure that's on that ascending aorta. And additionally, if the aorta does get too dilated, where it's at risk of rupturing, then surgical intervention is taken to try and remedy that abnormal dilation. However, there are no current treatments that are aimed to try and reverse this abnormal dilation of that aorta. So in order to do that and understand how the disease progresses further, we need to understand how the extracellular matrix degradation affects the mechanics of the aorta. So there are a lot of things that we do know and some things that we don't. For example, we can understand that the passive mechanics of the aorta due to elastin loss. However, the active mechanics, like how it contracts and dilates due to this elastin loss, aren't heavily researched. Additionally, we do know that this elastin degradation does contribute to changes in smooth muscle cells, also known as SMCs, and this can cause changes in SMC phenotype as well as cell count loss. Additionally, we know that piezo-1, a mechanosensitive ion channel, does directly relate to SMC contractility. And so that gave us motivation to try and explore piezo-1 for its implication in ATAA disease progression. So then going from there, we will be using an enzyme called elastase to enzymatically degrade elastin fibers to simulate the ATA disease, as well as using a YOTA1, which is an agonist for that piezo-1 channel. So we're able to simulate that channel and understand how it affects ATA or its progression in ATA, and then as well as using a molecule called LNAME. And this is because there are several cells within the aorta, but we want to just get that smooth muscle cell response. So we just want to isolate that response in general. So how LNAME works is it's a nitric oxide synthase inhibitor, and that means that it will inhibit any endothelial-driven responses that tell the smooth muscle cells to dilate, and then therefore allowing us to isolate just that smooth muscle cellular response. And then all of these tools combined will help us understand how that ECM degradation can lead to changes in aortic mechanics and then provide for possible therapeutics for ATAA in the future. So going into some of our materials and methods, we first start with an adult mouse, usually for about 2 to 3 months old, and we use both male and female, and then we remove the aorta and cut out 1 millimeter sections of the ascending aorta. And then for our control groups, those get directly mounted into our wire myograph, and the wire myograph is used to directly tell us the forces for contractile and dilatory responses. For our elastase-treated vessels, those go through 75 units per milliliter of elastase for 6 minutes, and that is then transferred to a stop solution of 100 micromolar of NaCl for another 6 minutes, and then afterwards it is transferred back into the myograph to do those contraction and dilation responses. And then just so we have a kind of baseline for how much each aorta can contract, we will give it, we will find its maximally induced contraction through using 5 milliliters of a 120 millimolar of potassium physiological saline solution. And then we would do that 3 times, and then in between each of those rounds, we were doing 3 washes of 5 milliliters of a physiological saline solution just to remove any of that excess KPSS in between. And then we go down to actually start the experiment. So that's when we add in either 10 millimolar of the iota-1 or 100 millimolar of the L-name, and to just kind of explain those mechanisms a little more. So piezo-1, this channel here, it's typically stimulated by mechanical stretch, but since we won't be doing that to stimulate it in this experiment, we use iota-1, which is an agonist for that channel, and it directly binds to the receptors on the channel, which allow it to open and let in that influx of calcium. So it converts that mechanical stretch into chemical and electrical signals to then tell the SMCs to contract. And then going into the L-name pathway. So what happens first is that an endothelial nitric oxide synthase within the endothelial cells interacts with an amino acid called L-arginine, and that converts those interaction into nitric oxide, which then can tell SMCs to relax. However, when L-name is added, that inhibits that whole process, which instead can allow the SMCs to contract. So we're seeing a different response by inhibiting endothelial cells from telling SMCs what to do. And so moving into the actual experiment, that's when we will be using a dosage response for contracting with phenylephrine, and that is done in 1 to 10,000 nanomolar increments. And then after that is done, we will wash 3 times with PSS to remove any of the remaining phenylephrine. And then before the contractions experiment, we have to calculate something called an EC50 value. And the EC50 is the halfway maximal contraction due to phenylephrine. And that kind of just gives us a point to allow the cells to dilate down from something. So instead of allowing them to dilate down from baseline, we kind of like pre-contract them so we can understand how their ability to dilate. And then for the dilation experiment, we will also be doing a dosage response with acetylcholine, doing that in still the 1 to 10,000 nanomolar increments. And then after that, we wash 3 times again with that PSS to remove any remaining acetylcholine. And then we will calculate an IC50 value, which is similar to EC50, but just the halfway maximal point for dilation due to acetylcholine. Then moving into our results up here. So this is for our just control group, so nothing added, just the aorta itself. And then for our elastase-treated aortas compared together. So here, this is the contraction and then also the dilation response to both. So we can see like very clearly the large difference between elastase-treated vessels versus our control. So we can say that their forces are like heavily diminished. One thing we can also note, though, is that the EC50 and IC50 values are very similar between both groups. So this kind of gives us an idea that the signaling between cells is still intact. However, that mechanical ability to contract and dilate has been diminished with the elastase treatment. And then we can go into our L-name and iota-1 responses. So L-name, just to remind you, it inhibits the eNOS pathway, and then allowing SMCs to further contract more in both the contraction and dilation response. So that's what we expected to see. And we did see that in our L-name alone. We can see here the max contraction it gets to is about 6 versus the control up here is at around 4. So we did see increased contraction in our contractile group. And then also in the dilatory response, we can see here that in the control, they did dilate versus with adding L-name, they were contracting for most of the time, which is what we expected to see. We're still seeing that similar trend, though, of however, the elastase group is still like significantly lowered. So we were able to conclude that removing the EC-dependent dilation with L-name does not rescue SMC contractility in elastase-treated aortas. And then one thing we also did note with the L-name group was that the EC50 values were significantly different from one another. And we think that this could be due to increased receptor accessibility. So degrading those elastin fibers could allow the drug to access the receptors more readily, and then also more increased SMC coupling. So SMCs are signaling with each other better to contract. And then also could be due to a decrease in tissue structure as well. So degrading that elastin could change the tissue structure. And then moving on to our Yoda-1 group. So just to remind you, the Yoda-1, it is a piezo-1 agonist. So it will stimulate that channel, allowing for influx of calcium. And so we expected to see an increase in contraction in both, increase in contraction for a contractile group and an increase in dilatory forces. However, the contraction was about the same between the Yoda-1 and the control. So we didn't really see much of a difference there. And then the same trend as our previous groups, we're seeing that elastase group like still having those like significantly reduced forces. So from there, we concluded that activating the piezo-1 does not alter the contractile or the dilatory responses in the control and elastase-treated aortas. And moving on to the images we see up here, we wanted to kind of get an idea of how the aortic wall changes after elastin loss. So from the control is this image here. And then this is for elastase-treated groups. So we have DAPI stained in blue for cell nuclei. And then in red, we stayed for the elastin fibers. We can clearly see that there's much less red here. So we know that the elastin fibers are degraded. However, we can also see kind of a blurring and a more intensified signal of the DAPI in the ELA treated groups here. So that kind of led us to believe that there could possibly be some damage or structural change, like cellularly as well, to these cells due to the ECM degradation. So we wanted to further investigate that as well in the future. And then also something that came up was our reduced forces in our KPSS data for our elastase-treated vessels. And so we wanted to ensure that the cells were still viable. So we wanted to, in the future, do a live dead staining to confirm cell viability. And then all these findings together are highly relevant to ATA, understanding how ECM degradation leads to changes in the aortic wall to further understand ATAA disease progression. Yeah. And I would like to thank all the members of my lab for coming out and supporting, and then all the mentorship they've given me throughout the summer, and thanking my PI as well, and my Bayer mentor. Thank you so much. Any questions? I would definitely say at the beginning it was very difficult because I was looking, it's really small, and I was like, I have no idea like what I'm looking at. Like there's a lot of like different like vasculature like surrounding the heart, like a lot of different vessels too. So it was initially hard to see where the aorta was, but I felt like I learned quickly how to like be able to remove that and then be able to continue the experiment. So yeah, definitely is like very fast-paced work environment with that. Definitely, yeah, because the mice initially like they're like anesthetized. However, if you don't get to them fast enough, then their heart starts beating in the aorta. You can't really use it anymore because the cells are kind of dead. So you have to be fast about it.
The wire, mile is
So the wire myograph, what we do is we like have the aorta and then we put it on like 2 pins, and then we are able to stretch it to like a physiological length based on like the pressure readings there. And then from the acetylcholine and the phenylephrine, those are the things that are allowing the vessel to contract and dilate. And the wire myograph is essentially like measuring those forces and giving us that data back. It's very little. It's very little. Thank you.
for some closing remarks, then thank you. It has been a pleasure. Always look.